Hey everyone, welcome back. We're diving into something pretty interesting today. You know how we always think immobility and blood clots, like they just go together?
Right, like it's a given.
Well, turns out there's more to the story. And it starts with, believe it or not, bears.
Bears.
Yeah, bears.
Yeah.
Specifically brown bears that hibernate for months. They're basically immobile, but they don't seem to get clots. So we've got these two research articles you provided. One from Science, Immobility-Associated Thromboprotection is conserved across mammalian species from bear to human, and then another, Sleep Like a Bear, from Insights.
Perfect.
So let's break this down. This could actually have some pretty important implications for how we manage our patients, especially those who are immobile for various reasons.
Absolutely, and you hit the nail on the head. It's not just about bears. What's really fascinating is that this protective mechanism, the one that seems to prevent clots during immobility, it's not unique to bears.
Oh, really? So you're saying it's also in humans?
Exactly. Researchers found it in people with spinal cord injuries, even healthy volunteers who are on bed rest for studies. So this points to a fundamental process, something biological that might be happening across species.
Okay, hold on. So we know immobility usually increases the risk of venous thromboembolism,
Yeah.
VTE. But now you're saying there's this protective mechanism too. How does that even work?
Well, think about it. Bears are inactive for months during hibernation. Their blood flow slows down. You'd think they'd be riddled with clots, right?
Right, you'd think so.
But they're not. And the key seems to lie in their platelets.
Platelets, the tiny blood cells that help with clotting.
Yeah, those. During hibernation, the bear's platelets become less reactive, meaning they're not as quick to clump together. And they release fewer of the substances that trigger inflammation, the stuff that leads to clot formation.
So their platelets are still there, they're just kind of chill.
Exactly. Like their clotting system takes a break along with the rest of their body. And what's even more interesting is there's this specific protein involved called HSP47. Yeah, think of it like a facilitator for clotting. It helps this key clotting factor called thrombin attach to platelets. And that kicks off the whole clotting process.
So HSP47 is kind of like the matchmaker, bringing thrombin and platelets together.
You got it. And not only that, it also interacts with neutrophils, those immune cells that release nets.
Nets. Remind me what those are again.
Right, those are neutrophil extracellular traps. They're like sticky nets that can trap red blood cells and other things, which can also contribute to clots.
Okay, got it. So HSP47 is a busy little protein.
It is. And what happens during hibernation? Well, get this. HSP47 levels in the bear's platelets plummet.
So less HSP47, less thrombin attaching, less neutrophil action. Basically less clotting.
Precisely. It's like the bears are naturally disarming their clotting system by reducing this HSP47, and that's where things get really interesting for us, clinically speaking. I mean, imagine if we could understand this mechanism, maybe even manipulate it somehow.
We could have a whole new way to prevent clots in our patients, especially those who are immobile. That's huge.
Huge. And remember, this isn't just about bears. The research on immobile humans showed the same trend. Less mobility, lower HSP47 levels.
That's wild.
Yeah.
So it's like nature's giving us clues about our own biology here. But okay, let's step back for a second and think about what this actually means for us, for our patients. What are the key takeaways here?
I think the biggest one is it challenges the way we've always thought about immobility and clots. It's not just a straightforward immobile equals clot equation. Our bodies, just like those bears, they might have a natural way to protect themselves.
So there's nuance to it. It's not as simple as we thought.
Exactly. But as you said, this is where things get really interesting. What if we could harness this natural protection? What if we could develop drugs that, do the same thing as this HSP47 reduction?
We could change the game for patients who are at high risk. People recovering from surgery, those with limited mobility, even people on long flights. Think about the possibilities.
We're talking about a potential revolution in how we manage VTE risk. But gotta be cautious here, this research is still early. We need more before we start changing clinical practice.
Of course, more research is always key. So we've got this fascinating link between immobility, blood clots, and this HSP47 protein. What else did they find? Let's dig into those specifics.
Let's do it. Welcome back. Let's get into the details of those studies we were talking about. The one from Science. Immobility-associated thromboprotection is conserved across mammalian species from bear to human, and the Insights article, Sleep Like a Bear. They both give us a lot to think about, especially how this whole HSP47 thing actually works.
Okay, yeah, let's break it down. So with the bear study, How did the researchers even figure out that immobility was linked to reduced clotting? Like, what did they actually do?
Well, they took blood samples from a bunch of brown bears, 13 to be exact, both when the bears were active and during hibernation. And they used this technique. It's called thrombolestography.
Thrombo what?
Thrombolestography. It basically measures how fast and how strong blood clots.
So they were seeing if the bear's blood clotted differently depending on whether they were, running around or sleeping it off in their dens.
Exactly. And the results were pretty interesting. They found that during hibernation, the bear's blood clotting slowed down a lot, especially the part that's driven by platelets.
Which suggests that something specific is happening, right? Like something triggered by just being immobile.
Right, and that's where HSP47 comes in. To really get down to the nitty-gritty, they did this thing called mass spectrometry. It's a way to analyze all the different proteins in the bear's platelets.
So they could see which proteins were there and how much of each one.
Yep, and when they compared the proteins from hibernating bears to active bears, guess what? HSP47 stood out like a sore thumb.
It was way lower in the hibernating bears.
Oh yeah, like way lower. 55 times lower on average in the hibernating bears compared to the active ones.
Wow, that's huge.
It was the biggest difference they saw, which made them think, okay, HSP47 must be a big deal in this whole immobility and clotting thing.
So they had all this data from the bears, but to really prove that connection, they needed more, right?
Right, they had to make sure HSP47 was the real culprit, so they moved on to mice. They actually created these special mice, genetically engineered so their platelets couldn't produce HSP47.
Wait, they engineered mice with no HSP47 in their platelets. That's pretty cool.
It is. And then they put these mice through a procedure that kind of simulates what happens in humans when you get reduced blood flow, the kind that can lead to clots.
So they were basically setting the stage for clots to form and then seeing what happened with and without HSP47.
Exactly. And what they saw was the mice without HSP47 in their platelets, they developed way fewer clots, and the clots were smaller too.
Okay, so now they got evidence from bears and mice that HSP47 is definitely involved. But how does this apply to humans? We don't hibernate after all.
That's where the InSight study comes in, sleep like a bear. They looked at humans who were immobile for different reasons. Some had spinal cord injuries, and some were healthy volunteers in those bed rest studies.
So a mix of folks, immobile for different lengths of time and for different reasons.
Right. And guess what they found?
They saw the same HSP47 thing.
Exactly. Lower HSP47 levels in the immobile people. And the longer they were immobile, the lower the HSP47 got.
Wow, so this really does seem to be like a universal response to immobility, no matter what's causing it or what species we're talking about.
It's a pretty clear pattern, and it points to something fundamental going on. It seems like immobility kind of flips a switch and tells our bodies, hey, let's tone down the clotting for a bit.
Right.
And HSP47 is a big part of that.
This is fascinating. So we see this link. Immobility leads to less HSP47, which means less clotting. But how did that actually work on a cellular level? Like, what are the nuts and bolts of that process?
Yeah, let's dive into that.
Oh, so we've seen that HSP47 is like super important for making clots. And when we're immobile, our bodies naturally lower that HSP47 to prevent too much clotting. But how does that actually happen? Like, what's the mechanism at play?
Yeah, so one of the coolest things they found in that Science article, the one about, the immobility protection across species, they saw that HSP47 basically acts as a dock for thrombin on platelet surfaces.
So it helps thrombin attach to platelets.
Yeah.
Which makes sense because we know both of those are needed to form clots.
Right. And the researchers, they figured out that if they blocked HSP47, they actually used a couple of different ways to do it, like those special mice we talked about that didn't have HSP47, and even some drugs that block it anyway. They found that when they blocked it, the amount of thrombin that could bind to platelets went way down.
Okay, so less HSP47, less thrombin can attach. Less chance of a clot forming. It's like breaking a link in the chain, right?
Exactly. But remember those nets we talked about? The sticky traps from neutrophils?
Yeah, those nets, they can trap red blood cells and stuff, adding to the whole clot formation mess.
Right. Well, it turns out HSP47 affects those too. The research showed that it activates those neutrophils, makes them more likely to release those nets.
So HSP47 is involved in, multiple steps of the clotting process. It helps thrombin attach to platelets, and it makes neutrophils go crazy with the nets. That's wild.
It is. And when you take away HSP47, you're essentially hitting the brakes on both of those processes that lead to clots. And the Insights article, Sleep Like a Bear, they really hit on how this relates to humans. They saw the same thing in those immobile folks, the spinal cord injury patients, the bed rest volunteers. The lower HSP47 meant fewer of those platelet-neutrophil interactions, so less nets overall.
Wow, so our bodies are kind of amazing, huh? We have this natural system to protect ourselves from clots when we're immobile, and HSP47 is like the control knob for that whole thing, protecting both platelets and neutrophils.
Absolutely. And it really flips the script on how we usually think about immobility in VTE, doesn't it? It's not just a one-way street to clots. There's this whole balancing act going on.
It makes you wonder, though, if we have this built-in protection, why do immobile patients still get clots?
Yeah, that's a great question. And the thing is, in a lot of cases, it's not just immobility. There are often other things going on, surgeries, injuries, underlying health problems, and those things, they can kind of overpower that natural protection.
So it's like the protection is there, but sometimes it gets overwhelmed.
Exactly. And that's why this research is so exciting. We might be able to use this knowledge about HSP47 to, give that natural protection a boost.
Like make it stronger so it can handle those other challenges. It's mind-blowing to think that studying bears hibernating could lead to new ways to treat or even prevent clots in humans.
It really shows how powerful basic research can be. You never know where the next breakthrough will come from.
So where do we go from here? What's next for this research?
I think a big focus is going to be on developing drugs that specifically target HSP47. The science studies showed that there are some small molecule inhibitors of HSP47 that actually worked pretty well in mice and even in human cells. So there are already some promising leads out there.
That's awesome. Any final takeaways for our listeners, especially those who are seeing patients every day?
This is a great reminder that our understanding of clotting, it's constantly changing. And it's not always as simple as it seems. Like with immobility, there's more to the story than just immobility equals clots.
And for all those medical residents listening, imagine a future where we can actually check a patient's HSP47 level to assess their risk of clots, or even use these HSP47 inhibitors to stop clots before they even start. That's the future of medicine right there.
It's a future that might be closer than we think.
Well, this deep dive has been incredible. We started with sleepy bears and ended up talking about a potential revolution in VTE management. Who knew?
It just goes to show sometimes the answers to our biggest medical challenges are hiding in the most unexpected places.
If you found this deep dive as fascinating as we did, we highly recommend checking out those full research articles. Immobility-associated thromboprotection is conserved across mammalian species from bear to human from Science, and Sleep Like a Bear from Insights. And let us know in the comments what other medical mysteries you want us to explore. Disclaimer and bibliography in the description.